This innovation is presented collectively as BrainBiochem, a communication interface bridging the gap between health preservation and treatment for multiple sclerosis. The innovation is radical in that for the conventional allopathic health industry, it introduces a novel lifestyle, biochemistry, and genetic risk factor assessment program with an associated nutritional support and nutraceutical intervention regimen, pioneered at a local academic institution for general global application in patients with multiple sclerosis, and eventually a series of mental and neurological conditions. The innovation is incremental. It provides a scalable platform for communication and coordination of assessment and intervention modalities, and it joins a variety of health risk assessment, risk mitigation, health surveillance, and health intervention services. It involves a system innovation for health insurance. This industry usually operates on actuarial data for disease management. By offering them a cost-effective health assurance and surveillance system, this innovation offers low risk and a good return on investment. The innovation is also modular in that it provides a viable framework for the otherwise unstructured and mostly self-funded wellness approaches to multiple sclerosis by creating a structured and objective health preservation and surveillance program with specific prompts for timely medical intervention. To explain why this innovation has developed around this particular disease entity, I need to provide you with some basic background on MS and what makes it so unique. Multiple sclerosis is a demyelinating disease of the central nervous system. It has inflammatory characteristics, and the insulating or protective cover of nerve fibers, called myelin, is damaged in the brain and spinal cord. This disrupts communication and causes a wide range of physical, mental, and even psychiatric problems. The World Health Organization in 2008 confirmed that MS is one of the most common neurological disorders and the most common cause of disability in adults. 60% can no longer walk within 20 years, with major implications on their quality of life and financial costs to society. Life expectancy is not reduced appreciably, which means that it affects adults during the most productive time of their lives. There's also a serious lack of information about the resources available to address it. Following the original description of a few cases by Jean-Martin Charcot in 1868, MS now affects approximately 2.5 million people internationally. Europe, America, Southern Canada, Southeast Australia, and New Zealand have the greatest prevalence, affecting middle and high-income groups. South African statistics are somewhat unreliable. The Multiple Sclerosis Society of South Africa suggests that there are only 5,000 people with MS in our country. Yet even conservative estimates, based on well-established epidemiological data, suggest that there should be at least 65,000 people affected. The disease primarily afflicts white, Indian, and colored people in our country. Multiple sclerosis may present with a variety of signs and symptoms. An interesting feature is that those parts of the nervous system that are most active also appear to be most vulnerable to demyelination. The athlete becomes paralyzed in the legs, the violinist loses function of their arms, the academic develops memory problems, and so on. Apart from the cruel irony, this also points to a wear and tear or supply and demand component issue in the pathophysiology of MS. There are a certain number of fixed risk factors related to multiple sclerosis. These are important when determining the diagnosis, but are not subject to modification really. More importantly, there are several well-substantiated risk factors for MS onset and progression, many of which go completely unheeded and unaddressed, even though they are all readily reversible with a verified subsequent reduction in disease severity and progression. And with that, we mean significant recovery potential. The present situation in South Africa in particular complicates a cohesive effort towards improving outcomes in multiple sclerosis. In 2013, there are less than 100 neurologists in South Africa split 50-50 between public and private sectors. The diagnosis of multiple sclerosis is not always clear. There is no known cure, so the diagnosis may not be pronounced immediately. When it is, most patients interpret this as a sentence to inevitable disability. Disease-modifying drugs, which may well reduce the number of episodes of relapsing forms of multiple sclerosis, are not always indicated, do not always work even when they are, do not prevent long-term disability, most require injection, many have debilitating side effects, and they may cost in excess of 10,000 Rand a month. New drugs appear continually on the horizon. However, there is a fundamental problem that continues to escape the attention of those doggedly pursuing a cure. As shown in our extensive research, apart from the obvious episodes of destruction of myelin, which characterize MS, there is also invariably underlying issues of suboptimal myelin synthesis, maintenance, and repair. MS inevitably appears to be the product of a double whammy, both an increased loss and an impaired pathway of manufacture and recovery of myelin. Neither immunosuppression therapy nor even the prospect of stem cell therapy can overcome the genetic and biochemical barriers to myelin repair. Planting more seedlings to replace the ones that are trampled cannot compensate for not watering or fertilizing them. The fallacy of most MS therapies is that they focus entirely on restraining the immune system, that is to prevent myelin destruction, while completely neglecting the identification and mitigation of well-known risk factors and nutritional, biochemical, and genetic abnormalities that compromise myelin maintenance and repair. So we chain the pit bull, but we never fix the picket fence. Not surprisingly, disease-modifying drugs do not seem to improve quality of life appreciably, although they do in the form of a reduction of relapses. But this is achieved at great expense and with many side effects. To illustrate the pervasive preoccupation with immunosuppression, in 2005 a group of highly motivated South African MS patients, again working on the assumption that disease-modifying drugs were all that were needed for recovery, eventually managed to force medical aids to pay for disease-modifying drugs. Unfortunately, the picket fence remains neglected. Few of us are asking why, and no one seems to be willing to pay a fraction of the monthly cost of a disease-modifying drug to get it fixed. We anticipate that this approach will be an invaluable solution to many MS patients who do not justify DMDs, or even those who do. Scientific support for our broken picket fence model is found in several studies. In this large Canadian study of MS sufferers who did not use DMDs, 15% actually improved spontaneously, whereas more than 50% did not deteriorate over 1 or 2 years of follow-up. Many appear to have focused on reducing their own risk factors instead. It begs the question, where should the emphasis really be? So where are the discrepancies, and how may these be addressed? Well, in summary, relapses in MS are a white matter problem, myelin. However, long-term disability is proven to be a grey matter problem. Neurons are eventually lost by cell suicide. There is funding for DMDs, but none for identifying and mitigating the risk factors. While DMDs may reduce the number of relapses, they do not prevent progression. And yet we and several others have shown definitively that addressing known risk factors reduces progression, even if there continues to be occasional relapses. The challenge there is that months and years of dread of relapse lies trapped in the heart of the MS sufferer. There's a great sense of disempowerment and frustration, tremendous misinformation, and an almost fad-like pursuit of health, but no cohesive plan for health preservation based on the patient's personal risk profile. So, from a conventional medical perspective, indeed, the best strategy is to identify those who deteriorate faster and to target the progressive stages. However, in the meantime, the common and completely reversible risk factors and deficiencies that impair myelin maintenance and repair every day should be identified and addressed to the point of being objectively normalized, and with these normal levels maintained over time. During the course of our research, we have discovered that for only about 1,000 Rand, which is inexpensive compared to the MRI scans and DMDs, many of the major, common, and reversible risk factors for MS can be identified. Many of these can then be addressed effectively through simple lifestyle, dietary, and specific functional supplementation strategies. The key objective is assessment, individualized support, verified improvement, long-term surveillance, and follow-up. So given this background, what would the ideal situation be? What would bring change through radical, incremental, systematic, and modular innovation? Obviously, greater awareness amongst healthcare practitioners and healthcare recipients is paramount. From experience, though, we know that personal communication with colleagues and patients by means of direct patient-related reporting is more readily incorporated in subsequent practice. It is the best way to educate colleagues. Then, timely diagnosis for the purpose of intervention, or even preliminary functional interventions during times where the diagnosis is uncertain. There must be prompt identification and mitigation of all reversible risk factors and deliberate reversal of all deficiencies using focused lifestyle, biochemical, and genetic risk assessments. Then there should be the development of personalized strategies for health maintenance. Long-term health surveillance by means of serial biochemistry and disability assessments, and ongoing health coaching. There should also be a prompt for effective intervention at times that function deteriorates rapidly or relapses occur. How might this be achieved? Well, the BrainBiochem innovation consists of 3 parts: connection, that is involving the interested parties; collaboration, informing them about what is wrong and how to fix it; and consolidation, developing and maintaining a long-term health maintenance and surveillance program. The first step is connection, establishing a platform. This includes making personal contact with a healthcare recipient. We envisage doing this by means of a dedicated call center that offers appropriate client relations management and obtains informed consent for both the clinical services and the research conducted through this platform. The professional contact component includes establishing and maintaining an active and efficient referral network with participating health practitioners and the means to record and exchange data effectively, and with preservation of patient confidentiality. The next stage is collaboration. This involves enrollment and coordination of the so-called RAFA regimen. Following the initial referral and enrollment, we do a lifestyle, biochemistry, and genetic risk factor assessment in consultation with the patient's healthcare practitioner. Thereafter, there is a regular process of proactive communication with prompts for regular follow-ups of disability and biochemistry levels, the reappraising of relevant risk factors, and referral to the patient's healthcare provider in the event of fluctuating or deteriorating health. Finally, there is consolidation, and this includes the ongoing reporting of relevant information to the healthcare practitioner and the recipient at a level where they're able to include it either in the professional care or self-care. The initial regimen selection is personalized as needed in response to serial blood chemistry assessments. The reason for all this is that certain components like iron, vitamin B12, and vitamin D may require alternative dosages or delivery methods to achieve the desired results. Further testing may also be required in response to persisting abnormalities. In cases where cholesterol or homocysteine levels remain elevated, additional strategies may need to be employed. For this reason, biochemistry needs to be assessed at least twice a year initially, with revision of the regimen and addressing of risk factors as needed. If there are changes in health status, these prompt further inquiries, assessment, or referral. As soon as we detect the need for clinical, psychological, or biochemical assessments, the healthcare recipient is advised to visit the healthcare provider. BrainBiochem does not envisage practicing telemedicine as is currently under question by the Health Professions Council. As soon as a clinical, psychological, or biochemical abnormality is detected, the healthcare recipient is referred to their respective healthcare provider. Also note that BrainBiochem is not in opposition to appropriate use of disease-modifying drugs. BrainBiochem engages fully in all the resources that are available. However, the emphasis lies in supporting the essential and objective measures that improve quality of life. So how do we plan to achieve all this? What are the parts that are needed for this innovation to be successful? For starters, the program must be credible, and that depends on being backed by appropriate science and evidence. Professor van Rensburg has researched and published extensively over the past decade, and the RAFA regimen is the product of this research effort. Confidence in this research lies in the fact that the University of Stellenbosch and the National Health Laboratory Service have actually participated and are joint owners of the patent. There needs to be validated lifestyle and risk assessment, and there's a tool to achieve this that has been pioneered by Professor Kotze of Genomics, which has also been validated in several studies and is linked to a comprehensive and robust database. There must be reliable biochemical markers that are both sensitive and specific enough to afford confidence that disease progression is less likely. In our experience over several years, normalization of these factors achieves the greatest level of confidence. In very rare instances where deterioration persists in spite of these values being normalized, further genetic and biochemical tests have typically provided answers and are the basis of ongoing improvements and refinements of the regimen. To meet the need for determining the genetic risk factors, Professor Kotze has established an evidence-based and cost-effective panel under a demyelinating diseases gene screen. This identifies the genetic risk factors associated with general metabolism and the production of myelin. The information obtained by means of this panel is a permanent investment in the patient's medical record. It is able to guide personalized supplementation and also addresses several other health issues and may guide the use of medication, including blood pressure medication, cholesterol-lowering medication, issues of hormone replacement, and even treatment of breast cancer and depression. There needs to be a validated disability assessment score, and there is, in the form of the EDSS. However, because it may be impractical at times to refer patients to healthcare practitioners for hands-on assessment, there is a need for an online questionnaire. In April 2013, a web-based method of capturing disease progression using an electronic version of the EDSS was validated. The online version is already being implemented on our system. There needs to be evidence that a registry-based service is effective for patients with multiple sclerosis, and again there is. The patient population affected by MS resembles the UK patient population fairly closely, and as such there is every reason to believe, together with our existing experience during the research effort, that this experience will be the same in South Africa. There needs to be an online database that provides efficient patient data capture, effective electronic communication, interactive web-based interfacing, compliance with health information portability and protection, flexibility, and affordability. The TeamDesk online database has been in use since April this year, and it meets nearly all our present functional requirements. Although it is designed in such a way that minimum technical input is required, it is envisaged that some specialized information technology support will be required in the initial stages. Ultimately, this platform may serve as a prototype for the development of a proprietary system. There needs to be a cost-effective solution to the biochemical and genetic risk factors that are identified, and there is. The RAFA regimen, which stands for Restoring Actual Physiological Homeostasis Approach, has been published by Professor van Rensburg. Difficulty in obtaining all the components in appropriate quantities and convenient packages eventually led to the decision to develop a patented product line. This was also to enable health funders to support the program, which they would not be able to do through self-medication strategies. The RAFA regimen is patented by the University of Stellenbosch, the innovators, Natrx, and the National Health Laboratory Service. The income from this royalty supports the services offered to patients and the ongoing research efforts to improve its composition. The product needs Trade and Medicines Control Council approval as well. We have trade approval to sell it in South Africa, and we're expecting Medicines Control Council approval soon. Ultimately, to be successful, this service needs to be viable, and it is. Most health services, pharmaceuticals or products are independently funded, and each focuses on its own profitability. In contrast, the commercial relationships in this innovation, both by virtue of shared patent and due to unique non-competitive, independent revenue streams related to each component, have innovated a sustainable ecosystem in which the MS patients are the greatest beneficiaries. For example, the university has access to state and private funding for the research related to MS and the ongoing refinements to the regimen and management protocols. Therefore, patients do not need to fund this through inflated drug costs, as is the case for most commercial pharmaceutical patents. The genetics and biochemistry services have the benefit of great economy of scale in that the tests are not unique, which means that they are typically less expensive, covered by health insurance, and can be offered at preferential rates to private patients. The product manufacturer has several other products, which means that the infrastructure for producing the product is shared, and therefore the cost of the product is lowered. The product is also designed specifically to be suitable for funding through medical aids, and yet still affordable for those who are uninsured. The unique patent and royalty structure of the product fosters ongoing collaboration between the parties and actually cross-subsidizes some of the testing and assessment components. This makes it more affordable to the patient and ensures that the regimen and product selections are optimal for them. In anticipation of entering the international market, the product has already been registered with the World Intellectual Property Organization. As the system has been designed to be scalable, the potential international market is potentially enormous. The composition is such that there are no barriers of entry into the EU, Canada, or the USA. The involvement of the University of Stellenbosch as a joint patent holder adds credibility to the project and assures appropriate ethical oversight. The university, by means of the Human Research Ethics Committee, has approved both the research itself and the use of the RAFA regimen in multiple sclerosis patients as part of a commercialization of the platform, now called BrainBiochem. One of the greatest benefits is that with proper informed consent given by patients, the data gathered through this program may be published and be of benefit to the greater medical and MS populations at large. A key factor to the success of this program is having access to a network of like-minded healthcare providers who are willing to support the program and the multiple sclerosis patients who are participating in it. HBO and diving medicine have a lot in common with MS. There's a history of the use of hyperbaric oxygen, and also the neurological deficits related to multiple sclerosis are in some ways similar to those of decompression sickness. So practitioners in diving and hyperbaric medicine are skilled in neurological assessment and have a passion for their work and are familiar and comfortable with communicating long distance. Moreover, the interest of the local hyperbaric and diving medicine practitioners has already been polled, and there is commitment to being involved. Finally, there needs to be a pool of patients, and in our case, there's already a pre-existing source of patients by virtue of the 10 years of ongoing research. In fact, as many as 100 patients who are still actively using the RAFA regimen are only too grateful that a convenient product line is on its way. As such, upon release of the product, which is anticipated by February 2014, there is an immediate long-term customer base of more than 100 individuals. As reported by the World Health Organization, it is well known that patients with multiple sclerosis seek information and appreciate quality communication. Uncertainty is the enemy of outcome related to their illness, and the experience of the researcher is that the grounded, practical, evidence-based, empowering approach is extremely well received by MS patients. What remains to be done For the innovation to be brought to its fullest potential is that the communication platform which already exists in semi-professional form now be brought to full commercialization and efficiency, together with expanding professional and public awareness. In terms of the client interface, the platform and communication tools will be structured into a health coaching, health surveillance, and health intervention component, making navigation and participation intuitive. The shift from disease suppression to health preservation brings about significant positive psychological shifts in patients with MS. With that, I would like to close with the words of the neurologist who first described multiple sclerosis, Jean-Martin Charcot. If you do not have a proven treatment for a certain illness, bide your time, do what you can, but do not harm your patients. I believe that this innovation represents the spirit of his recommendations.